morning, everybody. Let me introduce you the ERC Hy-Gate Research Project. HyGate is the acronym for Hydrophobic Gating in Nanochannels, understanding single-channel mechanism for designing better nanoscale sensors. Alberto Giacomello, Associate Professor of Fluid Dynamics at the Department of Mechanical and Aerospace Engineer of Sapienza University of Rome, is the principal investigator of this strongly multidisciplinary project. Good morning, Professor Giacomello.
Good morning, Silvia.
Would you like to tell us a little bit more? So, what is the project HyGate about?
Sure. So, Project HyGate aims at bringing an engineering perspective on a fascinating biological subject: how our cells communicate between each other and with the outer world. This happens through minuscule switches called ion channels, which allow the selective passage of ions through the cellular membrane. These ionic currents carry information throughout our body, for example, neuron firing in our brain and heartbeat, which keeps us alive. So you immediately see how important this is. So our research is about understanding some basic mechanism through which these switches work in order to be able to engineer bio-inspired nanovalves and nanosensors.
And can you tell us a little bit more about what you mean by bioinspiration?
So scientists are by now able to make nanometer scale pores, drill a hole which is 1 billion times smaller than a meter. However, this artificial pore cannot yet match the incredible capabilities of biological ion channels, which are capable of manipulating molecules precisely at these scales. For example, some ion channels are capable of letting large ions through the membrane while rejecting smaller ones. To use an Italian analogy, it is a sort of pasta strainer that is capable of draining spaghetti while retaining water. So recent research has shown that ion channels are able to carefully control bubble formation in their interior in order to effectively switch on or off. So these extraordinary capabilities of our cell switches explain why, as engineers, we are interested to learn the secrets that nature has devised along millions of years of evolution. This is bioinspiration.
And what application do you foresee from this research?
So, understanding how biological switches work will first of all help doctors fix when they don't work, which is the cause of several diseases, for instance, some severe heart conditions. It might also help to devise new, less invasive ways to achieve anesthesia in surgical operations. On the engineering side, learning from nature, we hope to be able to design new nanoswitches which could be used in smart membranes for water purification, for drug delivery, and for building new nanosensors.
What is the engineering contribution to this project?
So as engineers, we are using advanced simulation tools, which allow us to describe in a quantitative manner what happens within these nanoscale switches. In particular, we use molecular dynamic simulations to reveal the function of a given protein starting from its structure. This approach bridges scales which are either too small or too large for current experimental techniques. At the moment, we are simulating how bubbles form within these tiny channels, switching them on and off, and how different parameters can influence their operation, such that we can learn how to control them.
You say we. Who are the members of the team?
Thank you for this question. This is really the core of the project, a strongly multidisciplinary research team formed by young engineers, physicists, and biologists working together at Sapienza University. We have a lot to learn from each other, starting from different jargon we use. It's a really inspiring and fun experience. Here is a recent picture of the team members. I invite you to see the video presentation in order to virtually meet all of them.
And who is supporting this research?
So this large research effort would not be possible without the support of the European Research Council, ERC for short. This project is planting seeds which fruits will be visible in some time from now, and as you appreciated, requires a large team. So I'm really thankful to this European institution for giving me this unique opportunity to follow a new risky path. I would say a vision, which can bring benefits to all of us in the medium term.
So thank you very much, Professor Giacomello, and good luck with your research.
Thank you very much, Silvia.